This is the Illinois Nutrient Loss Reduction Podcast, episode 38, Soil Soldiers, Microbes, Fighting for Your Soil Health. I'm University of Illinois Extension's Todd Gleason. Today, we'll talk about microbes and the communities of microbes that are working within soil to improve its health, that of the plant, and of water quality. Stacy Zuber will help us kick things off. She's the Illinois State Soil Health Specialist with the Natural Resources Conservation Service, or NRCS. I caught up with her and asked her to tell us a little bit about herself, and then to explain just exactly what soil health might mean and why it's important to farmers and landowners.
All right, I am Stacy Zuber. I am the Illinois Soil Health Specialist with the USDA Natural Resources Conservation Service. So really my job is working on promoting soil health across the state and really trying to promote cover crops and no-till and other conservation practices.
How do you define soil health?
The official definition for soil health is the continued capacity of a soil to function as a vital living ecosystem for plants, animals, and humans. And I really think from that definition, the most important parts of that are really that we're looking at soil functions. We're looking at the processes that are happening within that soil. And then the living ecosystem component of that is a really huge part of it, because a lot of those functions and processes are really being done by the living organisms within the soil, our microbes and macrofauna and different types of organisms within that ecosystem.
Why is it that farmers, landowners should care about those living microbes, should care about the ecosystem?
Within the soil, we get a lot of benefits from the soil that it provides to us, having to do with nutrient cycling and having to do with capturing water and being able to store that water within the soil. We want to prevent erosion. There's a lot of different functions within that soil that are really important for the productivity. Our crop production for farmers is really dependent on the functions within that soil. So we really want to support them as much as possible. And the best way to do that, or one of the best ways to do that, is by having a soil microbial community that can really effectively do those processes and functions. And so we want to make sure that we're giving them the best environment for them to live in, and we're feeding them and giving them a fuel so that they are able to do those functions for us and make that soil more productive and more beneficial for the farmer.
What kind of microbes might we encounter in the soils of Illinois, and what are their roles?
Well, we have bacteria, we have fungi, and then, those are the ones that most people think of first and foremost within the soil. And a lot of what their roles are as decomposers, where they are breaking down plant residues, they are breaking down the root exudates that the plants are giving off. They're cycling nutrients through those. So those are our smallest that are out there. Then there's other organisms like protozoa and nematodes that are really involved in population regulation, where they are actually acting as predators and grazers within the soil, where they will eat those smaller organisms, the bacteria and fungi. And so that is really important for cycling of nutrients, because as those organisms, this protozoa, the nematodes, consume the bacteria and fungi, they are actually cycling nutrients, and nutrients like nitrogen that are stored within those bacterial cells, those fungal cells, when they get eaten by the protozoa or nematodes, some of that nutrients gets released back out into the soil. Some of it gets left over. The predators don't use all of it up. And so then that is released into the soil. And often that's happening right around the plant roots. So there's this process that's called priming. There's a priming effect where the plant roots actually can give off sugars and root exudates that attract the bacteria in. Predators will follow them and cycle and consume the bacteria. And then that leftover nutrients is right there by the plant roots, which is a really interesting way that we have that interaction between plants and microbes. So those are really what we're focused on for the microbes, those smaller ones. There's a number of other ones out there, But I do want to point out on the nematodes, it's important, sometimes they get a bad name because there are some that are pathogenic and problematic for our crop producers. But there's a lot of them out there, the vast majority of them, that are actually very beneficial for cycling of nutrients. And we really want them out there because they do a lot of those functions, a lot of those processes that are really important for productive soils.
Can anything be done to expand or maintain the microbial communities?
So the key thing with being able to increase the microbial diversity within our soil is thinking about them as living organisms. Just like us, they need a habitat, a home to live in, something that supports them, gives them the right temperature, the right moisture, the right soil pH. They need food to eat. So we want to, basically, the best way to do that is to make it so that it's very conducive, very supportive for them to survive in that soil and thrive, so we can protect the environment. So we want to protect the soil structure. So if we can minimize disturbance of the soil, if we can have a soil cover, by whether that's by having residues remaining on the surface, or by adding cover crops in there that can provide a living cover on the soil surface, during the normal bare time of the year. So we want to make sure we're protecting that habitat for them. So that gives them a place where they are able to live. And then we also want to feed them. And we want to give them not just one type of food. We want to give them a wide variety of foods. And we can do that by incorporating more different crops into our crop rotation, and maybe by also adding more diversity into our system by adding cover crops. And having a mix of cover crops can really help as well, because all of those different types of plant species within the soil are going to give off different types of fruit exudates and different types of food. And so when we have more diversity in the plants that we're incorporating, that can create a lot more diversity in the food sources, and that supports a wider variety of microbes. And then another way we can do that is by integrating livestock. If we add more above ground animals into those systems, they're also making a different type of food source for those microbes. And so then that also helps create a more diverse microbial community.
Stacy Zuber is the Illinois State Soil Health Specialist with NRCS. Angela Kent, as it happens, is a professor of natural resources and environmental sciences at the University of Illinois, who's been studying how to maintain and expand those microbial communities, the ecology of the environment in which they thrive. I asked her to tell us a little bit about the research she's been doing.
Sure. So my group is interested in studying microbial contributions to the nitrogen cycle quite often. And in particular, we want to help agricultural systems have sustainable nitrogen cycling processes. And what we zeroed in on is the contribution of the plant host as an ecological driver of the microbiome and its functions. So we're exploring different plant genotypes to see to what extent they control the recruitment of microbes with beneficial or detrimental nitrogen cycling processes. And of course, we want to increase the beneficial nitrogen cycling processes like nitrogen fixation, and we want to decrease nitrogen cycling processes that detract from environmental health, things like nitrification and denitrification in ag systems. So some of the exciting research that we have going on right now is the realization that in corn, we have perhaps bred out some of these sustainable nitrogen cycling processes. So we went back in time and looked at teosinte, maize's ancestor, and discovered that teosinte can actually suppress nitrifiers and denitrifiers. So it can suppress these detrimental nitrogen cycling processes, and that has been lost in modern maize lines. And so one of the things that we're doing is we discovered the genomic regions in teosinte that contribute to those processes, and we're trying to move those into modern maize. So we have some near isogenic lines that contain those genomic regions that suppress detrimental nitrogen cycling processes by microbes. And then the other aspect that we're interested in is working with Fred Belo's lab. We're interested in microbial inoculants that promote soil health and soil nitrogen provisioning. So nitrogen fixers, but then also some other additives that contribute to soil health. And then we've got an experiment in the field this summer where we're stacking some of these traits. So we're inoculating with nitrogen fixing microbes, and then we are hoping to keep that nitrogen in the rhizosphere by adding those inoculants to the nitrification inhibiting corn. So we want to provision nitrogen and keep it in the field.
How do you see this research being utilized moving forward in the crop breeding area for selection?
So I have a crop breeder collaborator who's very, very interested and very on top of that. But we're actually funded in part by the Illinois Nutrient Research and Education Council, which is funded by corn growers. And so we have specific objectives that were of interest to them. So we're making test crosses right now where we're making hybrids that contain the genomic regions that lead to the suppression of nitrification and denitrification. We're going to make sure that these capabilities don't depress yield, because we know that that's important too. And that was a concern of the corn growers when this project was reviewed. So we're moving right to yield trials pretty soon here, next summer.
Do you have any advice on how to improve and maintain soil microbes?
Crop rotation is really important, and I think anything they can do for adding organic amendments would also boost the yield too. Cover crops to help retain nutrients also would be pretty beneficial as well.
Well, some of that to-do list is fairly simple, like crop rotation, and other things on the list not so simple. Angela Kent from the University of Illinois is doing some research to try to figure out what else can be done and how effectively and efficiently it can be incorporated into modern farming operations. Operations, in fact, like Terry Davis's, who's from Roseville, Illinois. He's been no-tilling for 35 years, and in fact, he says he does work to maintain the microbial communities within his soils.
Yes, we have been. By the usage of reduced tillage, we've made the addition of cover crops into our rotations to help accelerate that, and we have modified timing and application of fertilizers to try to allow the microbial populations to thrive as much as possible.
Let's talk a little bit about the soil improvements you think you've seen over time since you started with the conservation practices. For instance, what were the first things that happened after you went no-till?
The first thing I noticed was and it was a detriment at the time, the tie-up of nutrients as the soil made that transition from oxidation of nutrients through tillage to biological activity releasing nutrients from the soil profile.
And once that was completed, did it release those back for the crop to use?
Yes, we did get everything back over the years. It took about 3 years for that to occur. I learned a lesson from that and have since then, when running new farm ground, taking the opportunity to do some inline tillage to continue to loosen that soil profile up, but not to turn that soil profile over, so that we allow the oxidation of the organic matter to happen through biological activity rather than through mechanical tillage.
Anything change in the way water flows to the soil profile?
Yes. Water infiltration has improved immensely. Water profile moisture retention has improved significantly. to the extent that in a bigger rain event like I had this last weekend, which in my area right here, we had between 3.5 and 6 inches of rain over a 48-hour period. And where normally you get into tilled farmland this time of year, that the surface will seal over and that water will shed off, where the no-till fields, that water usually stays on the fields, and you see some ponding that you might not have saw otherwise, just because that water is not leaving the field, but staying there now.
It has to do with root penetration and the stability of the size of the aggregate of the soil?
Both of those. We have noticed significant increase in soil aggregate size, and we have much more infiltration due to earthworm activity producing on an average 20 to 30 burrows per square foot. So we have a very porous sponge-like soil surface now, which allows that water to infiltrate rather than run off. But in large events like this, it takes that profile 24, 48 hours to be able to filter that water down through the profile, whereas before it just ran off.
Has your need for inputs changed?
It has over the years. My nitrogen usage is the main objective that we have tried to work with. My phosphorus and potassium levels on my owned ground and the family farm were in pretty good shape before I made the conversion. So I hadn't had to make many changes there. And on rented farm ground, even low rental rated rental farms, that we found that basically putting on a maintenance plus maybe about 10 to 15% extra has been adequate to completely satisfy the need of the growing crop that year.
You have more than 3 decades of experience with no-till. How long have you been introducing cover crops into the farm?
Cover crops began 6 years ago. Was my first foray into purposely seeding a fall crop for the sole purpose of cover crop, with the determination of termination in the spring. We had been planting some wheat previously, but and then double cropping beans behind it, but its purpose had not been as a cover crop per se, but as a cash crop at that time.
Did it change or make additional improvements, or possibly things that didn't go as well with water infiltration, the stability of the soil, need for pesticides, the earthworms and such?
As I said, with the addition of the cover crop now, we experienced this year, I'm getting such a good soil coverage in the spring due to the increased soil residue that's staying on the surface, that the cover crop on top of that is creating a mat of vegetation decaying on the soil surface, which unfortunately in a spring like this year means cold surface soil temps. And the germination of the soybean crop especially, I did note big differences in that this spring. Just a few degrees temperature change. Had a little frost damage one evening around Mother's Day, and that was solely caused by the soil surface being covered, that the heat didn't radiate back up off the soil surface as readily as it did off of tilled fields. And some neighboring fields did fare better than I during that time period, or even in my own fields, I'd see spots where we'd removed the cover, turning traffic and just bare soil, that the beans did fare better through that. Just a few hours of cold weather made a difference to those this year. But I think we'll get it back this summer, because those same soils are going to remain cooler this summer when we get into the high heat and that will reduce the stress level on the plants in late summer.
Given your experience, what are the things that producers should do to improve soil microbial health?
First thing, reduce the number of trips off the field with the mechanical tillage. Secondarily, to speed up the process, sizing of the residue early on seems to be a benefit. Long term, the reduction in denitrification products I've seen has made a big difference in my microbial activity. As I said, I do time my nitrogen application rather than in the fall, but in the spring, just ahead of planting. We lay down what we think is going to be needed and then come back in side dress depending on what we think the season's going to give us as far as increased opportunities for yield.
Terry Davis is a farmer from Roseville, Illinois. He's been no-tilling for some 35 years. You've been listening to the Illinois Nutrient Loss Reduction Podcast. This has been episode 38, Soil Soldiers, Microbes Fighting for Your Soil Health. The program was produced in conjunction with Illinois Extension Watershed Outreach Associates, Jennifer Jones and Rachel Curry. I'm Illinois Extension's Todd Gleason.